in this lecture, we'll talk about QSAR, that is Quantitative Structure Activity Relationship. And QSAR is a method of ligand based drug design. Ligand based drug design, yani when the receptor structure is not known, but the ligand structure is known. And not just the ligand structure, but many different things about the ligand are known. Like, for instance, the activity of the ligand is known. Okay, so if we do not know the receptor structure, but if we know the activity of the molecules of the ligands along with the structure, then we can resort to using QSAR. And QSAR is one of the most, next to docking, I think, and maybe next to PBDD, or maybe alongside PBDD. QSAR is one of the most widely used methods of computer-aided drug design. And not just that, it is also an accurate method of predicting the binding affinities or whatever is the activity that we have. If we are using MIC values or IC50 values, okay, so those values can be predicted. So it is a very accurate method of predicting the activity values. Okay, and that is the reason why QSAR is very widely used. Okay, and from amongst the different methods of QSAR, 3D QSAR is the most widely used. Okay, so QSAR is of different types, and from amongst those different methods, 3D QSAR is the most widely used method. Okay, and we had talked about artificial intelligence few lectures back, right? So we said that in artificial intelligence we use mathematics, more of mathematics and equations, whereas in drug design we use physics, right? So here in this QSAR, we are going to talk about physics and then we are also going to talk about mathematical equations. Okay, so QSAR is the basis of artificial intelligence, we can say, right? So QSAR is nothing but application of artificial intelligence. So initially, in the initial years, artificial intelligence was not known. So QSAR was known. But now we can say that QSAR is an application of AI, or in other words, AI is an application of QSAR. Maybe we can interchangeably say these things. Okay. So now we are going to talk about physics and about some mathematical equations in QSAR. Okay. So QSAR is a technique that allows to use statistics, okay, mathematical equations. So it's a technique that allows to use statistics to lower the trial and error factor in drug designing. So in pharmacophore-based drug design and in this thing, docking, right? We talked about how the ligand is binding to the receptor. But now we are not going to talk more about how the binding is going to happen. We are going to talk about how statistics can be used to find out the effect of substituent on the physicochemical properties of the molecules and hence the activities of the molecules. Okay, so in one of our previous lectures, I had explained to you all the difference between SAR and QSAR. In SAR, we talk about how the substituent change leads to change in the activity. But in QSAR, what we do is how the change in substituent changes the physicochemical properties and how does that affect the activity. Okay, so here we talk about the physicochemical properties in addition to talking about the activities. So what we are doing is when we change the substituent, what is happening to the physicochemical property and what is happening to the activity. We are checking both of them at the same time. So in other words, what we are trying to do is we are trying to derive an equation between the changes in physicochemical properties and the changes that happen with the activity of the molecule. Okay, so this is what is quantitative structure activity relationship. Okay, so it uses statistics and that helps us in drug design. So QSR modeling is widely practiced in academy, industry, and government institutions around the world. So it is a very widely used technique. The fundamental assumption of QSAR is that variations in the biological activity of a series of chemicals or molecules that target a common mechanism of action are correlated with the variation in the structural, physical, and chemical properties calculated through the software. Okay, in other words, what we are trying to say is for a given series of molecules that target a given common receptor, we can correlate the variations in their structure, physical, and chemical properties through software. Okay, so this is the same thing that I was talking about here. The variation in the structure leads to variation in the physicochemical properties, and that leads to variation in the activities. And this is correlated using statistics, okay, by the use of software. Structurally related properties of chemicals can be determined by experimental and computational means comparatively much more efficiently than the biological activity using in vitro and in vivo properties. Okay, so for instance, say suppose again, let us take the classic example that we have been talking again and again, naphthalene ring system. Okay, so unsubstituted naphthalene ring system will have, a particular amount of, say suppose we are talking of a physicochemical property, something like log P. Okay, so if we have a log P of, a certain value of log P for unsubstituted naphthalene, okay, and then we are trying this for some particular activity, like for example, LZ activity, non-steroidal anti-inflammatory activity, okay, analgesic activity. So this unsubstituted naphthalene has some log P, and that is responsible for certain activity. Okay. Now, what if we substitute this with something at position number one? That is going to change the log P, and that is going to give us new activity. What if we change some other substituent? For example, electron withdrawing डाल दिया था पहले यहाँ पे. Now we have a electron donating group substituted. So that is going to change the log P and hence the activity. Okay. Then similarly, we can talk about all of these different substitutions. Okay. And that will give us large number of molecules with different log P values and different activity values. So now we know the differences in log P and the corresponding differences in the activity when the structure changes, right? So we can derive equations using statistics. Okay. And we don't have to do it per se. Initial beginning when QSAR started, the researchers used to do manual calculations. But now we have softwares to do that for us, right? So we have the quantitative structure activity relationship, QSAR software, which we can use to do these calculations. So we can find out whether how log P is related to activity. So that means any substitution that we do, which will change the log P in a certain way, is going to change the activity also in that particular certain way, right? So this is how QSAR is derived. Okay, so structurally related properties of chemical can be determined experimental or computationally by means of comparatively much more efficiently than the biological activity using in vivo in vitro approaches. So what we are doing here is we are taking physicochemical property, we are taking the activity that is known from maybe in vivo or in situ or in vitro method, and then we are applying statistics to it using software. We are deriving equations out of it. Once we change the values for the equation by changing the substitution, we'll be able to predict the value of the final molecule. That is the use of QSAR. Okay. So a statistically validated QSAR is model is capable of predicting the biological activity of unknown chemical compounds of the same series and can be used before the synthesis and biological evaluation. Okay. So we have been talking about computer-aided drug design reduces the iterations, number of iterations. So this is an example. QSAR is also an example wherein we are using a validated, statistically validated model, and that can be used to predict the biological activity of unknown chemical compound, unknown molecule. But the problem is the prediction can be done within the same series only. So if I have derived the QSAR model for naphthalene, and then I start wondering if I can design some biphenyls. No, I cannot do that using QSAR. Okay, so for that, I'll have to resort to either pharmacophore based drug design or better docking. Okay, but for QSAR, this is a limitation. We have to do the predictions only within the given series. So, जो naphthalene based molecules है, we'll be able to predict activity only for those molecules, naphthalene series of molecules. Okay, and this can be done before the synthesis and biological evaluation. So we need not synthesize large number of naphthalene molecules in this example. Okay, we can just selectively synthesize a few molecules that have been predicted to be having good biological activity. Okay, and then we can after the synthesis of those molecules, we can go for biological evaluation of those limited synthesized molecules. That's it. This is the use of QSAR. Okay, so based on the calculations of descriptor values, structural representations are derived, and dimensionally based methods of QSAR are categorized into the following classes. So as I was saying, there are different classes of QSAR, of which 3D QSAR is the most widely used, right? But there are other different classes as well. So we have 1D QSAR, we have 2D QSAR, we have 3D QSAR, 4D QSAR, 5D QSAR, and 6D QSAR. So 1D QSAR, it talks about correlation of activity with properties, molecular properties like pKa, yeah, log P. Okay. 2D QSAR talks about correlation of activity with structural patterns, not with molecular properties, but with structural patterns like connectivity, 2D pharmacophores, without taking into activity the 3D representations of these molecules. And 3D QSAR overcomes that this particular drawback as well. 3D QSAR, it correlates activity with non-covalent interaction fields surrounding the molecule. Then we have 4D QSAR. 4D QSAR, it additionally, in addition to 3D QSAR, it will also include ensemble of ligand configurations in the 3D QSAR. So in 3D QSAR, we use just one single conformation of ligand, but here we'll use ensemble and a collection of conformations of the ligand, and that is 4D QSAR. 5D QSAR, it explicitly represents different induced state models in 4D QSAR. So 4D QSAR के आगे वाला step is 5D QSAR, wherein induced fit models are used. And 6D QSAR is further incorporate different solvation models in 5D QSAR. 5D QSAR का अगला वाला model is 6D QSAR, wherein the solvent model, the solvent effects are also taken into consideration. Okay, but needless to say, I'm repeating it, I think third or fourth time, 3D QSAR is the most widely used method from amongst all of these QSAR methods. So linear regression analysis. So I've just talked about using statistical analysis, right? So linear regression analysis methods are the most easily interpretable amongst the various statistical methods that can be used for QSAR. So linear regression analysis means what? A property X will correlate directly with property Y. Okay, if we plot a graph and we plot X on one of the axis and Y on the other axis, then we should have a linear correlation. So that is what is linear regression analysis. Okay, but not necessary that always will have linear correlation, right? We can have different types of correlations as well. Okay, and for that we'll use different methods of regression. Okay, so the following are the different regression analysis methods which are used for QSAR. One is obviously linear regression analysis. Okay, so these are the different examples of linear regression analysis that can be used. The other one is multivariate data analysis. So we have important examples like principal component analysis, principal component regression, partial least square analysis with a PLS method, genetic function approximation. So of all of these, PLS is widely used. So you all need not be stressed about learning all of these statistical methods, okay? But what you need to understand, what we all need to understand is QSAR is based on application of statistics to the physicochemical properties and hence activities. Okay, then we can have genetic partial least square analysis. Then we can have pattern recognition, linear regression analysis, wherein we talk of cluster analysis, artificial neural networks, k nearest neighbor approach, etc. ओके। एंड ये जो पैटर्न रिकॉग्निशन टेक्निक्स है ना, these are the ones that are widely used in artificial intelligence. Okay, these are the ones that are widely used in artificial intelligence. Obviously, these ones are also used, principal component analysis, PLS analysis, etc. And the linear regression analysis are also used, but artificial intelligence more widely it uses the multivariate data analysis and pattern recognition techniques, okay, as compared to QSAR. So getting back to QSAR, linear regression analysis is most widely used in QSAR, but other types of regression analysis can also be used. Okay, so let us talk a bit about 3D QSAR. So 3D QSAR, the classical methods of QSAR are based on on the regression analysis of the relationship between the biological activity of a set of homologous compounds and the descriptors with various description with various descriptors. For example, we may have activity plotted versus log P. Okay. And what we'll try to do is we'll try to set up a correlation between these. Okay. So for a given series, if activity and log P are linearly correlated, then for an unknown molecule from that particular series, we can always extrapolate and find out on the basis of a log P what would be the activity. Right. So that is how the usual classical QSAR methods are used. Okay, and the correlation equations of these relationships predict the activity of new analogs from the same homology group, right? But in addition to the classical QSAR, at present the method of 3D QSARs are widely used, and special methods of description of 3D distribution of ligand properties are used in these methods. Like for instance, we have the one of the most widely used methods called as COMFA. COMFA is comparative molecular field analysis, COMFA. And then we have COMSIA, which is another method. COMSIA is comparative molecular similarity indices analysis. Okay, so we have these two methods of 3D QSAR. One is called as COMFA, comparative molecular field analysis, and the other one is COMSIA, comparative molecular similarity indices analysis. So all of these, both of these utilize characteristics like steric characteristics or electrostatic descriptors, hydrophobic lesion descriptors, etc. Okay, so this is how the 3D QSAR differs from the routine QSAR. Okay, in that we have COMFA and COMSI as the two methods which use steric, electrostatic, hydrophobic regions around the molecules, and these properties are calculated in a very specific way. Okay, so for instance, I have been talking to you all about probe atoms, right? In previous lecture on physicochemical property analysis, we talked about probe atom of a certain diameter, right? 1.4 degree Armstrong and a certain charge. So what does that mean? Okay, let me just quickly explain to all of you how 3D QSAR is done. Okay, so is done is a regularly spaced hypothetical grid is generated. Okay, alongside a molecule. We have a say suppose we have a say this is aromatic ring OCH3. Okay, and maybe a OH group over here. So say suppose this is one of our design molecules. So what is done by COMFA-COMSIA is it will draw a regularly spaced grid alongside this molecule. Okay, and then a probe atom will be used at the intersection of these grids. Okay, a probe atom would be placed at the intersection of these grids, and then that probe atom will calculate all of these parameters, steric, electrostatic, hydrophobic region related parameters. Okay, so for instance, we can have a probe atom first. It will be kept at this intersection. Okay, अभी ये intersection से सबसे पास क्या है? We have the CH3 group, right? And CH3 group is not as such, right? CH3 is CHHH. So one of these hydrogen atoms is very near to this intersection. The other hydrogen atom is okay, थोड़ा सा distant. The third hydrogen atom is far away from this particular intersection, right? So this probe atom will measure all the properties for that hydrogen which is very near to it and that hydrogen is, which is kind of distant away from it. Okay, so let me draw one more intersection here. Okay, so see, now we have, अच्छा चलो मैं ऐसे draw कर देता हूँ H H and H CH3 group. Okay, so this hydrogen, all the properties of this hydrogen will be calculated by this particular probe which is placed at this intersection. When the probe moves on to this intersection, it will calculate the properties of this hydrogen. When the probe moves on to the next intersection, it will calculate the properties of the next hydrogen. When the probe moves here, it does not have anything in its vicinity. When the probe moves here, it will calculate all the properties for the carbon of the methyl group as well as the oxygen of the methoxy group, right? Carbon and oxygen of the methoxy group, all the properties for those atoms will be calculated by the probe atom when it is at this intersection. Similarly, when this probe atom will move to this intersection, it will again calculate few more properties of this oxygen. It will move to this, it will calculate few more properties of this oxygen and this carbon over here. When it will move to this position, it will calculate the properties of this oxygen and this carbon, right? So on and so forth. This probe atom will move all across and it will calculate the values. Okay, it will calculate the values for each of the atoms that are present in this particular molecule. And this is how 3DQSAR analysis is done. Okay, now whatever we want to study, for instance, we want to study log P, okay, and we want to correlate it to activity. So what will this method do? Compa, COMSIA, these will calculate all of these values and they will calculate the log P values on the basis of these kind of calculations. Okay, so ये जो probe atom है, which will have a diameter of something like 1 or 1.1 or 1.4 degree angstrom, and that will calculate the hydrophobic properties. Okay, now if we and the steric properties. If we want to calculate the electrostatic properties, we'll have to introduce the charge on that probe atom, maybe -1 or +1. So then that will calculate the electrostatic properties, so on and so forth. Okay, and on the basis of that, we'll have the log P being calculated, for example. Okay, and then that log P will be correlated to activity by the software. Okay, so these are the steps via which the 3D QSAR will progress. Okay, so it will place the molecule onto a regularly spaced grid. अभी ये grid जो है ना, that is also defined. So say this is 1 millimeter into 1 millimeter, or maybe 1 micrometer into 1 micrometer. So that will depend. Okay, the spacing of the grid. The more tightly the grid is spaced, the more difficult the calculation would be, the more time consuming the calculation would be. So that is the reason why many times the grid is kind of relaxed as well, so that you know the points at which the number of points at which the calculations are to be done would be reduced. Okay, so that is what is ridiculous. Yeah. Now this approach is widely used because of significant difficulties encountered with SBDD in converting calculated binding energies into real values of KD, KI, IC50, and so on and so forth. So yeah, this is something that we had talked about docking as well. Docking as well, we cannot calculate or we cannot correlate binding affinity with KD values or KI values or IC50 values. But with QSAR, that is possible. With QSAR, we can calculate the binding affinity in terms of KD values or KI values or IC50 values. Okay, so that is the reason why recently 3D QSAR models have been designed for the subsequent prediction of activity of compounds found in molecular databases or designed de novo, even when the target 3D structure is known. Okay, so nowadays, even if the 3D structure is known, see, if 3D structure of the receptor is known, we can go for SBDD, right? Like in SBDD, if we go for docking, it is not going to calculate all of these properties. So instead of that, even if the 3D structure of the receptor is known, the predictive ability of the 3D QSAR models is so good that this method, 3D QSAR, is still followed even if the receptor structure is known. Okay, so that is how 3D QSAR is very widely used nowadays. Okay, Now let us talk briefly about the chemical descriptors in QSA. Now we know that in 1890s it was Mayer and Overton who independently were working on toxicity of organic compounds, and they found that toxicity of organic compounds was dependent on the lipophilicity. Okay. Nowadays we know it very easily, but at that time this was a discovery for them in 1890s. Okay, so Mayer and Overton, they said the toxicity of organic compounds depends on the lipophilicity, right? In the basis of the study. And then we know the field of QSAR modeling was further founded by Hans et al. Right? The first name of Professor Hans was Corwin. So Corwin et al. With his studies, which he did in a very small lab, in his he did it in a very very small lab, in a very small facility. He used to work with his students, not just his students, but he also used to work with his students, and they used to study those different types of descriptors. Okay, so initial study was done by Mayer and Overton, but the field of QSAR modeling, it was developed because Hans and his colleagues and his students, they worked on different types of descriptors which were later on used for QSAR modeling. Okay, hence Hans is said to be the father of QSAR. Okay, although Mayer and Overton were the first ones to be studying QSAR per se, right? So they were trying to correlate toxicity to lipophilicity. In other words, they were trying to study QSAR, right? But it was the pioneering work by Hans and his colleagues and his students that led to the development of QSAR to what it is in today's world. Okay, so chemical descriptors are at the core of QSAR modeling, and so many different types of chemical descriptors reflecting various levels of chemical structure representation have been proposed so far. So these levels can range from molecular formula, that is 1D, one-dimensional descriptor, to the most popular two-dimensional structural formula related descriptors, or the three-dimensional conformational dependent descriptors, or even higher levels of descriptors, taking into account the mutual orientation, time-dependent diametric dynamics of the molecules, etc. Okay, so this is how chemical descriptors have cropped up after the studies of Hans and his colleagues. Okay, so let us have a look at few of the most important descriptors that are used in QSR modeling. So we have constitutional descriptors, we have electrostatic descriptors, topological descriptors, geometrical descriptors, and quantum chemical descriptors. Okay, so constitutional descriptors are molecular property related descriptors, like for example, molecular weight or the number of halogenated atoms, halogen atoms, okay, etc. etc. So that is constitutional descriptors. Electrostatic descriptors are the charge related descriptors. So partial charge, then we have charge surface area, etc. So these are electrostatic descriptors. Then we have topological descriptors, and again surface area related descriptors, and these can be connectivity indices as well. Okay, so we have different types of connectivity: zero, first order, second order connectivity, valence connectivity. Okay, so these are the topological descriptors. Topological is surface and connectivity related. Then we have geometrical isomer descriptors. Descriptors would be the solvent's accessible surface area, then molar volume, etc. Then we have quantum chemical descriptors. Quantum chemical descriptors, we have examples like dipole moments, quadrupole moments, polarizability, HOMO energies, etc. So quantum chemical descriptors are the descriptors which are based on quantum mechanical calculation. Okay, whereas the other descriptors are widely used in molecular mechanical calculation. Not that quantum descriptors cannot be used, but as is the case, quantum chemical descriptors are more widely used for quantum mechanical calculations, whereas molecular mechanical calculations, the other four types of descriptors are used. Interpretation and statistics, we can apply a large number of statistics to QSA. Now, this is something that I'll just run through because I do not want you all to be, absolutely stressed about the interpretation and statistical significance of the molecular model, especially because most of you won't be doing QSAR as a part of your studies, right? So we have many different statistical parameters which we can use to find out whether the given QSAR model, whether the QSAR model that we have developed is statistically significant or not. Okay. So we have standard leave one out validation. We have standard cross validation. We have bootstrapping validation. And finally, we can use no validation, okay, NV, to determine the R square value. R square value is nothing but the correlation coefficient. Okay, R square value is nothing but the coefficient of correlation. Okay, and all of these validations are used so that the equations that we get are able to predict the activity of the unknown molecule accurately. Yes, that is what is mentioned here. Cross validation by low O is one in which the compound is removed from the data set and its activity is predicted using a model derived from the rest of the data set. Okay, using an optimum number of components. Usually six is the default number of components. So similarly, we have different types of validation. So leave one out validation is one of the better methods of validation. And the last way of validation is no validation, wherein the actual prediction of the R square value and Q square value is done. The Q square value is nothing but cross-validated coefficient, Q square. Okay. And these calculations we can do manually as well as we can do them computationally. Okay. Then there is something called as R square predicted. R square predicted is the correlation coefficient for the predicted value. And the equation that we have developed for QSAR, what is the predictivity of that equation? That is determined by R square predicted. Okay. So that is standard deviation minus stress upon standard deviation. Standard deviation, all these are terms. We need not get into the details of these terms, but R square predicted is the one which helps us to find out how good would be the predictivity of the unknown molecule. There are other parameters as well. We have R square Pearson's correlation. We have F value, which is variance ratio. We have P value, which is significance level of the variance ratio. We have RMSE, root mean square error. Okay. And we have Q square value, which we have talked about in the previous slide. We have Pearson's R value. So these are the average values for which for all of these parameters. So when we are using QSAR, R square value should be as high as possible in between 0 to 1. So if the R square value is 1, the QSAR equation is validated. It is perfect. Okay. If it is 0, it is absolutely imperfect. F variance ratio should be as high as possible. P value should be as low as possible. RMSE should be as low as possible. Obviously, this is an error, right? So it has to be as low as possible. Q square value should be in between 0 to 1 as high as possible. The better the value of Q square, the better would be the predictivity of the QSAR equation. Pearson's R value should be in between 0 and 1. It should be as high as possible. Okay. So these are examples of uh, you know this is pharmacophore based alignment. Okay, so here we can see this is one aromatic ring, this is another aromatic ring, and then we have a hydrogen bond acceptor over here. Okay, and then we have other aromatic rings as well, but those are not a part of the pharmacophore. Okay, so this is uh, so these are considered as a sphere. This is this sphere, and then we have three features, that is R1, R2, R3. R1, R2, and I think this is R3. Okay, so that R is not visible over here. These two are definitely not the rings. So if we have three rings, one, two, and three ring, and the third one hollow sphere, which is indicative of acceptor, hydrogen bond acceptor, then that is the pharmacophore. Okay, so this is how we can visualize the pharmacophore. And here what we see are the contour maps which come out of COMPA and COMSIA. Okay, 3DQSAR, COMPA and COMSIA. Okay, so now these are black and white images, but they have G written over here, R written over here, so they are indicative of the color. So G is green, B is blue, R is red. Okay, and what do all those colors mean is also mentioned. For example, in this particular figure, yellow color and green color indicate regions predicted to cause unfavorable and favorable steric interactions with bulky substituent. Okay, so yellow means unfavorable, and so yellow is somewhere here. Okay, and where is it? Green indicates favorable. So this is green, this is green, this is green. So what does that mean? That means in this molecule or in the pharmacophore, we can have sterically bulky substitutions at these positions. Whereas if we have a sterically bulky substitution at the yellow position, that is going to be detrimental to activity. If we have sterically bulky substitution at the green position, then that will be increasing the activity. Okay, so that is what is meant by these contour maps. So we can always have those contour maps which are produced out of 3DQSAR equations and can use them for predicting the activity of unknown molecules. Okay, so that's it. We have finished off QSAR as well. Okay, and the criticism about CAD we have already talked about in one of our previous lectures. So we are done with QSAR.